Hello and welcome to the third video for GCSE Biology B5 and B6 revision. In this video we're going to have a look at antibiotics and painkillers, the development of drugs and monoclonal antibodies, looking at the production and some of their uses. To start off, I would like you to answer these three questions for the Do Now task. Please pause the video now to answer the questions. The answers are now on screen for you to mark your answers. We're now going to move on to looking at antibiotics and painkillers. So we will first look at the discovery of antibiotics. And a key person in this was this chap here, who was called Alexander Fleming. And he was a doctor working at St Mary's Hospital in Paddington, so central London, in the 1920s. And in 1928, he went on holiday. And he left all of his petri dishes on the side. When he came back, he discovered a mould growing. That looked a bit like this. And we have the petri dish that is here. The yellow substance is a bacteria that has grown on the petri dish. And this green part here is the mould that he discovered. This clear space around it, there was no bacteria here. And he found that this mould had killed all of the bacteria around it. This mould was known as penicillin. Unfortunately, it wasn't until 1942 that we managed to mass produce penicillin and use it successfully against various infections. But since then, it has gone on to save millions and millions of lives. This discovery is often known as a serendipitous discovery because it happened purely by chance or by accident. He wasn't meaning to find this mould, but by going on holiday and leaving the petri dish with the bacteria and the mould growing in it, he discovered penicillin, which gave us great hope for saving lots and lots of lives. Now we can use antibiotics for treating bacterial infections. So antibiotics can treat bacterial infections. We cannot use antibiotics to treat viral infections. So we cannot use antibiotics to treat viral infections. And the reason for this is that a bacterial infection, the bacteria are easy to get to. They are in the blood or in the skin. They are not in cells. So they are not in cells. A viral infection, viruses make us ill by entering cells. So our viruses are inside cells. So bacteria or antibiotics cannot get to them to destroy them. Now, if you've ever been ill and been to the GP or the hospital and received antibiotics, you would take them and you would start to feel better. We do have some important rules for when we take antibiotics. Our first rule is we must only take antibiotics for bacterial infections. We must only take antibiotics for bacterial infections. Second rule, we must always finish the course of antibiotics. We must always finish the course of antibiotics. The reason for these rules is to prevent antibiotic resistance. Now, antibiotic resistance is where bacteria are no longer killed by or destroyed by the antibiotics. This is because they have mutated. Because bacteria mutate at a high rate, they can very easily become resistant to antibiotics. The first bacteria that became resistant to penicillin after it first got used happened a few years after it was first mass produced. They become resistant very, very easily. Now, these are obviously rules for us taking them. There are other things we can do as well. So a lot of farmers will use antibiotics in their feed to help with cattle growth and stop them getting ill. That increases the rate of antibiotic resistance. So we can also add on to our rules a third one if we wanted to, which is reduce antibiotic use in agriculture. So we reduce antibiotic use in agriculture, and again that will help prevent antibiotic resistance. We're now going to have a look at painkillers. Now, painkillers are another type of medication. So they are a medication. They are a drug. And painkillers are used to alleviate pain. And painkillers can be taken at all sorts of times. They can be taken if you have a headache. So if you have a headache, painkillers can help reduce that, or the pain from the headache. You can take painkillers if you are feeling unwell, so if you have a viral infection. We have a virus within us here inside the cells. Painkillers can help with that. So if you have an illness, you can sometimes be in pain, and painkillers can help with the pain there. Or you can take painkillers if you are having surgery. So if you are having surgery, you can take painkillers. You can also take painkillers for any other type of pain as well. So for example, back pain or muscle pain, painkillers can help alleviate that pain. And importantly, painkillers do not get rid of the infection. So painkillers do not get rid of the infection. There are now 5 questions for you to answer. Please pause the video and answer the questions. You should now only be watching if you have answered all 5 questions. The answers are on screen now for you to answer the questions. We're now going to look at the development of drugs. And first up, we're going to look at some drugs that we derive from plants. There are 2 drugs that we need to know we can derive from plants. The first is aspirin, which is a painkiller. And we get that from the bark of a willow tree. Nowadays, aspirin is synthesized in a lab, so it's made in a lab, but it used to be derived from a willow tree. People were able to chew the bark of a willow tree to get the pain relieving symptoms. The next drug we'll look at is Digitalis. Now, Digitalis is a heart drug. Digitalis is a heart drug. It is used to correct arrhythmias in the heart, so dodgy rhythms of the heart. And we get that from the plant foxgloves, which is a nice purple plant. Nowadays, again, Digitalis is synthesized in a lab, but it is based on the chemical that is within foxgloves. We're now going to have a look at how new drugs are developed and the process that we go through. So we have numerous steps. Our first step is to identify a new drug. At this point, we might have thousands and thousands of drugs that we can think might be useful. We make that drug in a laboratory and we test it on cells and tissue. We test it on cells and tissues. This is to check for toxicity and efficacy. So toxicity, to see if it will kill the cells and tissues, and efficacy, to see how well it works on the pathogen or on whatever it is we want to be treating. At this stage, those thousands of new drugs that we might have thought of, we're now down to hundreds of drugs, potentially even less. Our next stage, we will test it on animals, particularly mammals. We will test it on animals, particularly mammals, and we do that to see how the drug interacts or works on a living organism. So we will see how the drug works in living organisms. We will use mammals because their physiology is very similar to humans. And from those hundreds of drugs we had first, we are now down to tens of drugs. Our next step, we will test on healthy volunteers. We will test it on healthy volunteers. And this is to check for side effects. We will check for side effects. At this stage, we may be down to potentially 1 to 2 drugs that are successful. Our next stage, we will test on people with the condition or infection or illness. So we will test it on people who the drug is for. And this is to check it works. Again, we might get maybe 1 drug that might potentially work, maybe 2. Our last stage, we do large clinical trials. And our large clinical trials are there to check that the drug works better than existing drugs, to check for the right dose, and to check that we are happy with it and any regimes we might need to go on. Some drugs require you just take it once a day, other drugs maybe 2 or 3 times a day. And the important thing about these clinical trials is that we have 2 groups. We have a group that is either a control group, so we'll have the current treatment regime, so they'll either have the current treatment or they will have a placebo. Now, a placebo is a substance that has no therapeutic value. So a placebo is a substance that has no therapeutic value. Other group who are not the control, either with the current practice or the placebo, will receive the new drug. Now, these trials are double-blind. They are double-blind, meaning that the researchers and the people taking the drugs do not know who is in which group, and that is to reduce bias. So double-blind means the researchers and the people in the trial do not know who is taking which drug, and that reduces bias. Our next bit we'll be looking at is drugs in the future. Now, a lot of antibiotics are no longer effective due to antibiotic resistance. So a lot of antibiotics are no longer effective due to antibiotic resistance. So we need to develop new drugs. And scientists are doing this by looking at plants again, just the same as how we got willow to give us aspirin and foxgloves to give us digitalis. We are looking at plants. And we are looking at plants to find their properties. We saw in the last video how some plants can produce antibiotics or antibiotic chemicals to fight off bacterial infections. They can also produce various pain relieving chemicals as well as hallucinating ones as well, which might have a pain relieving side effect. So we can get bacterial chemicals as antibacterial chemicals, or we can get pain relieving chemicals. We can get pain relieving chemicals. There was a lot of research going on into this of finding new drugs. There are now 5 questions for you to answer. Please pause the video now to answer these questions. You should now only be watching if you have answered all 5 questions. The answers are on the screen now. We're now going to have a look at monoclonal antibodies. First, we'll look at the production of them. So we have here a rat, and into the rat we will inject a substance. And that substance will contain the antigen. And it could be the antigen for a pathogen, or a hormone, or an enzyme. It can be the antigen for any particular thing. Inside the rat, we start to see an immune response. The B lymphocyte cells will produce antibodies. The B lymphocyte cells will produce antibodies, and these antibodies are specific to the antigen. We will then combine the B lymphocyte with a tumour cell. Now, a tumour cell divides rapidly, and this is important. Once we have combined these 2 cells, we have a resultant cell called a hybridoma. We have a cell called a hybridoma, and a hybridoma will produce antibodies, and it will divide rapidly. All of the cells that divide from the hybridoma will produce the antibodies and will also divide rapidly. And that means we can get a large number of antibodies very quickly. And these antibodies are all monoclonal antibodies. They are all monoclonal antibodies. And those monoclonal antibodies, mono means one, and clonal is for clone. So they are all clones of the same antibodies. They are all identical, or they are all clones. We will now look at some uses of monoclonal antibodies. Now, what we have here are one of the lateral flow tests that we're all using at the moment to test for coronavirus. And this works using monoclonal antibodies. Now we'll know that we place the sample here. So our sample goes into this gap here. This is known as the reaction zone. This is known as the reaction zone. Inside the reaction zone, we have some monoclonal antibodies for the coronavirus antigen. And these monoclonal antibodies have an enzyme attached. Now when the specimen moves up the strip, so up the test, if the antigens are present, they will bind with the monoclonal antibodies. Up here in our test window, we have what we call the test zone and the control zone. So we have the test zone and the control zone. Now in our test zone, we have the antibodies, or the monoclonal antibodies, for the monoclonal antibodies down here. So we have some more monoclonal antibodies. And these monoclonal antibodies have a dye attached to them. Now, if the two monoclonal antibodies will bind to each other, which they will do if they've been carried up by the sample, the enzyme will cause the dye to appear. If they do not remain at the test site or the test zone, because there are no coronavirus antigens, it will continue to move up and go past it. If it is positive, we will see a red line appearing in the test zone, telling us that the antigens for the coronavirus are present. We will always see a red line in the control zone, and that is to tell us the test has been successful. So it will tell us the test is valid, and a red line in the test zone tells us that the test is positive. Now, the same principle also works for pregnancy tests, where we're also able to use monoclonal antibodies in research. There is some thought that monoclonal antibodies can be used to treat cancer. And we can do this by delivering chemotherapy direct to the cancer cells. So we can use monoclonal antibodies to deliver chemotherapy directly to the cancer cells. There are now 4 questions for you to complete. Please pause the video now to complete these questions. You should now only be watching if you have completed these questions. The answers are on the screen now. Thank you very much for watching. Have a great day. Bye-bye.